First of all, I want to thank all the participants today for coming here and celebrating this wonderful event. And thank you for accepting our committee invitation and coming into the, spending a day here. I just want to say that David has reached a full circle today because he started his work with analyzing ribosomal structure and understanding ribosomal metabolism and its function. And he is still focused on it after 63 years. And I had the opportunity to continue some of the work, and I would like to share that with you today. And just to emphasize the point that we are still not finished. So, this shows structure of the ribosomes, a single unit of ribosome here. Ribosomes are ribonucleoprotein complexes in the human cells. They are the machinery to make proteins using messenger RNA. And the organelle is composed of two subunits. It contains a large subunit and a small subunit. Based on their sedimentation rates through the sucrose gradient, they're called 40S and 60S subunits. And they are essential for every cell because you have to make proteins to function. And they use the messenger RNA that is stored in the DNA. And they translate into proteins. A single unit of ribosome is structured out here. It contains a 45S transcribed unit that is and continues with the intergenic spacer. This is a single unit, and there are 400 copies in each cell, each human cell. So only this part is transcribed into an RNA, and that RNA is processed by some of the work that was done at Davis lab earlier and many others. Shows that it can follow 3 different pathways to separate the 3 RNAs, the 18S, 5.8S, the small fragment here, and the 48S. So it can follow one of these paths to finally separate all of these 3 and then associate with the respective proteins to make the large subunit and the small subunit. So our goal has been to try to understand in great detail what is the arrangement of these copies of DNA. There are 400 copies in each cell, and this raises a lot of problems because we know that when there is duplication of the genes in a cell or in a species during evolution, the genes do tend to diverge. And if you want to maintain up to 400 copies of cells, the problem is how do you keep them all of them the same? Because it will lead to natural mutations may diverge each copy, and that will have an effect on the function of the ribosomal RNA. So, and the problem is intensified today because we do not know the complete structure of all of these 400 copies on different chromosomes. The 400 copies are distributed on chromosomes 13, 14, 15, 21, and 22. So eventually we need to address how the diversity is avoided, or if the diversity arises, how do you manage with it, and how do you control the expression of these genes? We do not really understand these aspects. Suppose you accumulate a mutation, how it is spread across different copies, or how do you manage the effect of these mutations in the translation, which would eventually affect the translation of the proteins. Also, we'd like to understand there is evidence in the literature that there is loss of rDNA during aging. So this will reduce the capacity of these cells to produce proteins, and that may have additional effects. So we really need to work on those. In order to understand all of this, we need to understand this structure in the chromosomes and how it is regulated, and how do we control these effects of these mutations. The cell must have some sort of mechanism to avoid all these issues. So as initial work, what we wanted to do was to understand the structure of the ribosomal RNA locus on one particular chromosome. So we took a hybrid cell line, a mouse human hybrid cell line that contains human chromosome 21. And then we use a specialized technique called TAR cloning, that is transformation associated recombination technique. I will go into a little more detail in a few seconds. And we sequence using the latest sequencing technology, using long reads, which will produce up to 50 kilobase of reads in one stretch, and use that sequence that we generated to find any variants that are present in the rDNA, and whether those variants would cause any specific effect. And we can also use this information eventually to see, to estimate the exact number of copies in an individual or in a population, using some of the techniques that was developed by Jun Ding to analyze mitochondrial DNA, which is also present in multi-copies in the human. So the transformation associated recombination, or the TAR cloning, is basically a simple principle. So suppose you want to isolate this large fragment of DNA, hundreds of kilobases, as shown in red here, and we pick unique sequences on flanks on this TAR cloning target. We clone that unique sequence into a vector and use that as hooks. And with the help of yeast cells, we mix this clone hooks or mix it with total DNA that is high molecular weight, and then let the yeast do the recombination work. So what the yeast does is it finds its match for the hooks, and then it copies everything that's in between, and then rescues the DNA, and then constructs a circular molecule in yeast. And this vector also has a property that it can exist in the yeast cell as a circular molecule, and it can be moved into bacteria eventually, and then where it can be amplified, and you can collect the DNA and then do further analysis that we want to do. So this shows our strategy to isolate the rDNA here. On the top line here, it shows the a representation of the repeats on the chromosome that is present on chromosome 21. And the primers that we chose, the unique set of primers that we chose, fall just above the 18S start site. And we picked different fragments of DNA that can be cloned. And we cloned them into a vector that's shown here as one of the examples. For example, this yellow one is cloned behind the green one. The reason for that is that if the recombination begins here, it has to go through the whole repeat and then find the next repeat where it finds this fragment. So that way we can rescue everything that's in between here, from here to here. So we have a representation of the full repeat. And we also chose to make complements of these sequences and then clone them in different orientations. Because as we go along, I show you that not all the repeats are in the same orientation. This illustrates some of the features of the vector that we use. The hooks are cloned at this point, and this vector can be amplified in bacteria. And then when we are ready to do the transformation, we cut out this part which is responsible for amplification of bacteria, and then make this vector linear. And then we mix it with the high molecular weight DNA from the from the cell line that we are interested in, and then we'll let the yeast do the recombination work. And this cassette will help us to, when we move into bacteria, to amplify the product that we want to sequence. And in yeast, we use the history selection so the molecule can be isolated in the circular DNA. Before we did that experiment, we wanted to make sure that the chromosome 21 hybrid cell line had the right DNA. Here we probe the DNA spread, the cell line DNA on the slide, and then hybridize with human-specific centromere sequence. And then you can see a single spot, which is shown here in the spread. And this inset shows an expansion of that to illustrate the point. And this shows a merge between the DNA state and the expanded part. So this shows that this contains a human chromosome. And below this is a probe that we use from the spacer region that is downstream of the locus. And that shows the presence of the chromosome 21, human chromosome 21 DNA. So, but you can see that the rest of the mouse chromosome does not light up with this probe. Just the human DNA that lights up. So this shows you that the probe doesn't light up. It is not specific. It's specific for human, but it does not identify any of the ribosomal DNA from the mouse. So you can stretch the DNA on a slide, and then you can use different probes that's shown here in green or red, and then we can highlight the actual ribosomal DNA structure on a fiber in a technique called what is called a fiber fish. So where the DNA is stretched along a slide, and then you can look in the microscope after hybridization the different portions of the rDNA locus. What you see on the right side is the order that we expect if the hybridization worked in this way. The green signal is followed by the red signal, and there is a gap after that because there is no probe that was used in this region. So you see this dotted green, red, and the space that you see. So this is what is called a canonical arrangement of the ribosomal DNA locus. But if you look on the other side, further in other fields, you can see that some of this arrangement is changed. For example, here is a longer stretch of green signal, red signal, flanked by green signal. So this shows that there is rearrangement of the rDNA in the system, in this hybrid cell line. And we can quantitate that. The average length is expected to be the probe size, but you can also see the distribution on either side of the expected size. For the 7 kb, you can get as long as 24 kb stretches because it's a fusion of all the green signals together. Same thing for the red signal, as well as the gaps that we expect. So this shows that the arrangement of the rDNA locus that we isolated in the 13 clones that we sequenced using the long-range segment. And the top 6, all of them contain rDNA units up to here. Some of them contain 2 units. The sequence analysis shows that it is a full complement of the rDNA. In some cases, you see that the spacer is truncated, and that's shown in orange signal, orange color. Some clones have 2 repeats, 2 rDNA repeats that we have recovered, and these recombination has produced many of these clones and the hooks are in the right place. And but we also recover some unusual structures. In this case, there is only spacer region, and the spacer region is not in the same direction. If you notice the arrows, they are going in the opposite orientation. So this is the result of rearrangement in the order that is expected. This could have happened at some point during the isolation or in the original cell as well. So, but it's a proof that you can get alternate structures in the rDNA locus. Further, what we can do is we can stack up these reads on top of each other and see if there is any difference in the rDNA sequence. And that's shown in the next slide. One of the first things that we noticed when we did this was to show that in the database there exists a model reference sequence that was put together in 1990s by Gonzalez and Sylvester. And we noticed that there's a deletion in that particular sequence, about 2 kb. The beginning of the deletion is on the top here at coordinates 23,120 and ends at 25,120, missing 2 kb worth of sequence. So we see that all of our clones show that sequence across, and also there are some variations at the junction of this piece that's missing from the original reference sequence. And we think that the reason for the original reference sequence was missing this is because it is in a region of repeat. So when you do the assemblies, the repeats tend to collapse on themselves, so you cannot tell the difference between the two, the full repeat. So that's why it was missed. So that's one of the features that we were able to rectify in the existing reference sequence. Further, you can see that there is variation in this number of CT repeats in this region, and that's a variation that hasn't been known before. The important thing for us to know is whether there are any variants in the 18S and 28S region, which is the functional portion of the rDNA. You can see that in this one, in different clones, you can see the number of Gs here is different. And in this case, there is a G that is instead of an A. So these are the kind of variants that we can detect in our sequences. So we have been able to catalog several of these, and this shows an additional variation further downstream in the 45S coding sequence. We have cataloged these variants, and we looked at their distribution across the different features of the rDNA, and there are close to 300, close to 300 variants that we have been able to catalog. And then we find that the question now becomes whether these variants arose in the hypercell line or the original chromosome or during cloning. So we need to figure out what is the origin of this. So what we did was try to see if it present in the existing sequences that has been assembled in the human genome, 1,000 genomes, or in the deep sequence individuals. So there are three to four examples of high fidelity sequences in the database called platinum genomes. They have been sequenced in many different ways, and they contain high depth sequence, and then we can compare that sequence to the variants that we have here and ask the question whether they are represented in that particular deep sequence individuals. So we find that in the deep sequence individuals, many of them out of 235 here, for example, in the intergenic spacer, we can identify 108 of them can be validated. And also you can look at, this can be validated in all the deep sequence individuals, whereas 148 of them can be validated in any one of them, okay? So we know that these variants that we have isolated, we have identified are valid, and they should be distributed through the population in some way. So that needs to be worked out by sequencing more people, of course. And the variants can be broken down into either single nucleotide variants, or multiple nucleotide, or single nucleotides, insertions and deletions. So all of this becomes important during trying to figure out what is the effect of this function. If you take a global look at the variants, if the dark blue is indicated those variants that were validated in different portions of the rDNA. This is the 18S, 5.8S, and 28S. Majority of the variants fall into the 3 terminus of the 28S RNA. And if you go back and map those variants onto the secondary structure of the RNA, we can see that they fall into certain portions of the rDNA in what's called the expansion segments. These expansion segments are species specific. They have been accumulated through the evolution to support the function of the particular species during evolution. There is a core structure that is maintained through the evolution everywhere, and most of the mutations fall outside of the core structure, but they fall into the expansion segments. So this gives us a suggestion that the mutations are concentrated on the expansion segments, so they can, this gives the chance during evolution to accrete new functions, that it's possible to accrete new functions. So the current status is that we are continuing to isolate more of these rDNA units from chromosome 22 to see if we can enhance the number of variants and then verify them. And it has been facilitated by long reads techniques, because if you make short reads, it's very difficult. The computer programs are unable to assemble the whole rDNA units. We'd like to generate a catalog of variants and include the variants eventually to a chip or to sequence to identify in the big populations of studies. And also it will give us room to think about what happens with these variants when they're expressed, whether they have any specific effect. We can correlate with some of the features in the population studies that we are doing to see if a specific variant inherited through in certain people has any effect on certain traits. And this work was facilitated by, of course, the drive by David, and some of the sequencing was done in our sequencing core, and the long range sequencing was done with the collaboration with NITRI, and car cloning was done with our colleagues at NCI, Larry Allen, who are the experts in doing such kind of work. I think I will end here and thank everyone.